Hi, welcome back. This is chapter 7. This week we have chapter 7 and chapter 8. They're a little bit shorter than the first part of the class because now we're starting to put things together, fewer introductory things, more new and interesting things. Today we're going to talk about sex determination and sex chromosomes across many different species. So we're going to look at turtles, at butterflies, at mice, at humans, and the different way that we use chromosomes or other things to determine sex. Before you start, I think you should be comfortable with the definition for chromatin, which is something we're going to expand on. But basically that loose bit of DNA that is accessible, we're going to refer to it as hetero or euchromatin as we go forward, and I'll break that down what those differences mean to you. Fertilization, which is putting two gametes together, that results in a zygote, which is a fertilized egg. Transcription and translation come up in a few lectures, and I think I talked about them in chapter 7 too in passing. This is taking the DNA code and ultimately turning it into a usable protein. And finally, gene expression, which I think you should have down, but it's a word that comes up a lot in the next couple lectures. Today, I only have 2 parts for you for chapter 7 to talk about the X and Y chromosomes in humans and something called dosage compensation, because we've been pretty clear the fact that males have a Y, but we haven't considered the flip side of that, where females have 2 X's, and that means you have 2 alleles for every gene, where a regular human can exist with just one of each of those alleles. And so we're going to have to balance that out. I'll come back to that, how that works. So in animals and some plants, actually, sexes differ phenotypically. You can tell the difference between a male and a female by expressed characteristics. We tend to start out when we start using genetics in our everyday life thinking that those sex chromosomes are what completely determine the sex of an animal, whether they develop as male or female, but that's not always true. The sex chromosomes have some genes on them that are used to determine sex, but in many animals, it's more than that. In plants too, it's more than just there is a gene for maleness or there is a gene for femaleness. Development is complicated, and the development of the sexes is just as complicated as everything else. A little bit of history here in how we understood it. This is a butterfly, Papio protonator. It's a spangle butterfly, and it was one of the first animals that the sex chromosomes were understood. In these butterflies, the females are XX and the males are X with nothing else. There is no Y, so there's no chromosome with the gene for testes on it. And a lot of insects work that way. This is actually taken out of a paper for grasshoppers. Grasshoppers work the same way. Females have one more chromosome than males, and that results in the production of eggs or sperm. The male either passes on an X chromosome or a sperm with fewer chromosomes than the one that has the X in it. If you inherit fewer chromosomes from dad, you develop as male. If you inherit an X from dad, you develop as female in these. And this is called a protonator sex determinator model. This is Papio protonator. The Ligius sex determination model is based on the milkweed bug. This is the Ligidae, the family Ligidae. The females pass on an X, and the males pass on an X or a Y. This is what we as humans use. If you're a female human and you have 2 X's, you can pass on an X, and there's another side to this, or another X. Plus you pass on one of each of your autosomes. Humans have 46 chromosomes. You pass, right now, inside of the cells inside of your body, you have 44 autosomes and 2 sex chromosomes. If you have a Y, you have 44 autosomes and 2 sex chromosomes. Same thing. All humans have 46 chromosomes. If you are female and you have 2 X's, you pass on half of your autosomes and one of your sex chromosomes. And the only option you have for sex chromosomes is X. In your other eggs, you pass on half of your autosomes and one of your sex chromosomes. Either this 50% chance of that X, 50% chance of that X. If you are male and you have a Y chromosome, you pass on 22 autosomes and an X 50% of the time. Or the other 50% of the time, you pass on 22 autosomes and a Y. That's 50%. Males have two possible things they can pass on, X or Y. Females can only pass on an X. I can flip this, how I talk about it, and I can talk about the gametes that are produced. Females in an XXXY system, the Lygeus system, based on the milkweed bug, pass on X's into all of their gametes. So we say that their gametes are all the same. They are homogametic. Homo means same. Gamete means gamete. If you're XX, all of your chromosomes have 22 autosomes and an X. If you have a Y, you are heterogametic. Hetero means different. This guy passes on an X 50% of the time, a Y 50% of the time. Y's are actually smaller than X's. We can centrifuge these apart and separate the ones that are carrying X's and the ones that are carrying Y's. His gametes, they're all sperm, but some of them have an X and some of them have a Y. They're heterogametic. They're different kinds of gametes. What type of offspring results from him contributing a Y? And we did this in pedigrees. Yeah, I mean, if he passes on a Y, he has a son. If he passes on the X, he has a daughter. And in the XXXY system, the male is actually the one that's determining the sex of the offspring. If he passes on a Y, he's passing on the gene to develop testes in this case. Birds are a little bit different. Female birds are ZW. Males are ZZ. Males are actually the homogametic sex in this case, and female birds are heterogametic. Female birds have a 50% chance of passing on their sex chromosome Z and a 50% chance of passing on their sex chromosome W. Males have a 50% chance of passing this Z on, a 50% chance of passing this other Z on, but all of their gametes will have Z's. These chickens have 76 autosomes and 2 sex chromosomes. They have 78 chromosomes. That's a lot. And we'll talk about why chickens, I mean, you feel like we should have more, right? You feel more complicated than a chicken? Some days. Some of these chickens, some of the gamete, the number of chromosomes actually doesn't determine necessarily if you're really complicated or you have a lot going on. There's other things at play here. She is heterogametic. He is homogametic. What happens if the male contributes a Z to the zygote? Well, in this case, male chickens don't determine the sex of the offspring, so he always contributes a Z. What matters in this case, whether you have a hen or a cock, is a male chicken, a hen or a cock is going to be determined by if the female passes on a Z or a W. If she passes on a W, it's going to develop, the baby is going to develop into a hen. And this is something I want to ask you on exam 2. I want you to be able to recognize the difference between hetero and homogametic. This pattern, the ZW pattern, is common in reptiles. Birds are reptiles, and so a lot of the reptiles have a ZW system. Not all of them, though. We'll look at some later that absolutely use something completely different. Oh, I clicked through. So your book has a whole bunch of different model and chromosomal sex determination systems. What you're looking at here is if chromosomes are used to determine the sex of the offspring, there's a couple different patterns. There is, and we'll look at this, these are C. elegans. Their sex determination is going to be you are either a hermaphrodite or male. Drosophila, I'm been lying to you a little bit about Drosophila, so we'll look at their sex chromosomes are XX and XY, but the Y doesn't actually have testes on it. We're going to look at how they actually do that determination. Mice or you would go here. The determination of many mammals is XX, XY. Weirdly, moss, which is a kind of plant, also uses an XX, XY system like we do. That's weird, but some fish, their sex determination is based on the number of individuals of the opposite sex they run into. They can change during their lifetime. You've seen, what is it? His name is he ruins everything, right? But talk about it too. When Nemo, in Finding Nemo, when you can skip this if I'm going to ruin the film for you. You've seen it, right? So when in Finding Nemo, his brothers and sisters are eaten, and they go back and they hide in the sea anemone. Only Marlin and Nemo are left, like his dad and his son. But what should happen in that kind of fish and in many kinds of fish is, because there are no females left, Marlin, who would have been the biggest fish left, would have actually changed over to egg production and would have made it with Nemo, and they would have reestablished the population. So they didn't have to go to Australia. But that's not really a kid's movie. And then frogs in this case, like in many other herps, so amphibians and reptiles, they have a ZZZW system. Herps are reptiles and amphibians, cold-blooded ones. So I want to look at this idea of, with that segue, with this idea of aneuploidies, or abnormal chromosome numbers, and we'll look at humans for this, because we've been looking at sex chromosomes and then saying that you have 46 chromosomes. That doesn't have to be true. And there are a number of known aneuploidies. An means not, U means true, ploidy means number of sets of chromosomes. These are not the true number of sets of chromosomes. It's an abnormal number where you usually, in humans, have one more chromosome than expected. There is one case where you can have one fewer. Most humans have 22 pairs of autosomal chromosomes, that's 44, plus the 2 sex chromosomes, so one pair of sex chromosomes. When we look at these karyotypes, and we did this in other lectures, this is kind of laying out the chromosomes of a human. These are human chromosomes laid out in order by size so we can look at them. Down here, this 23rd pair is an X and a Y. And for this, there's 2 people here. Draw a line in between. For this other person, it's X and X. So when we look at the average wild-type human, we see that our options are XY or XX for our sex chromosomes. I want to talk about 2 of the possible variations in chromosome numbers that affect the sex chromosome. And these are not all of them, and I have some other variations in chromosomes as we go through the class. But these are aneuploidies, so not the true number of chromosomes that are affecting the sex chromosomes. And generally, the effect of having one too many or one too few chromosomes of the sex chromosomes is less devastating than of the autosomal. And I'm going to show you why in a little bit. I don't remember if it's this video or the one after, but it's this chapter. This is Klinefelter syndrome, and so this is a karyotype showing Klinefelter syndrome. In this case, we've lined up all the chromosomes, and you can see that this person has an XXY as their sex chromosome. That means their chromosome number is 46 plus 1. We refer to this as a trisomy. At one position, one type of chromosome, there is one too many. So this person has a trisomy of the sex chromosomes that results in a syndrome, many different symptoms, called Klinefelter syndrome. Generally, this results in being taller on average than people. We used to think it was tied to extreme aggression. Your book goes into a really interesting and long description of that, but that looked to be more, in retrospect, from sampling error. They were sampling prison populations and found that men with Klinefelter syndrome had a higher chance of being very aggressive. But it's sort of a biased situation. I have a prison population where men are already fairly aggressive because of many reasons. It was not quite because of Klinefelter syndrome. When we look at the general population, we don't see that correlation. This is Turner syndrome. Turner syndrome is having one fewer sex chromosome, one too few. That sounds like a count. This person is missing the other X or other Y. They have 46 minus 1 chromosome, so only 45. This is called a monosomy. A monosomy is here at this sex chromosome position, one of the chromosomes is missing. The only viable monosomy in humans is Turner syndrome. Turner syndrome results in it's a syndrome. It results in a series of expressions of problems. You probably have recognized Linda Hunt. She is the spokesperson for the Turner Syndrome Society, and she also has Turner syndrome. You can develop, and we'll talk kind of like hinting at where we're going to go. You can develop fairly normally, but you tend to develop short. She's a very tiny woman. They have, when you're born, you tend to have some webbing in between your ears and your shoulders. In the modern world, we remove that, and there's no evidence of it. We have plastic surgery. There are some fertility issues associated with this. There may be some developmental delays in learning abilities, but it's not drastic, which is pretty surprising considering you're missing an entire chromosome. Triple X syndrome, or triplo X, is where you have 3 X's. How many chromosomes does this person have? So normal wild type people have 46. This person has an extra X plus 1. I could also write that as 44 autosomes, XXX. Does this person, do you think, develop as male or female? This person develops as female. There's no Y. So probably female. It is possible to be XXXX or XXXXXX. There is a reason you can have extra chromosomes here and not fail to develop. There are recognizable developmental, recognizable symptoms from being triple X, but considering this is a lot of extra genetic material, it's not what you think. Also, just FYI, you do have your project coming up for chromosomal disorders or genetic disorders. If this is something that is interesting to you, just holy cow, turn on your safe search. I did not when I was trying to find this karyotype. Triple X actually will bring up all kinds of things. Didn't think that through. XXY will not bring up weird things when you search for it on Google. It's when you have 2 X's and a Y. So this person is 44 autosomes XXY. They're 46 plus 1. Did they develop as male or female? They have the Y, so they probably develop as male. This is extremely common in the human population. I actually have some examples I can show you where it's common in cats as well. As far as we know, there are no developmental issues that come from this. You can't tell until I do your genotype. So you might be XXY and not even know it. That starts to beg an interesting question. These different syndromes and conditions I've shown you have a whole chromosome worth of information, extra or missing, and yet they seem to develop fine. I promise you, if you were missing or you had 1 extra chromosome 1, you just would not develop. If you have 1 extra 21, this is what Down syndrome is. It's a trisomy 21, or 1 extra chromosome. There's a whole host of developmental and other symptoms that come from having 1 itty bitty extra chromosome. Why are these special? I want to take a little segue here and talk about how sex actually develops in humans. This is a model of the Y chromosome, and I cut the key out. I took it out of your book, but I popped the key in the middle of this. During embryonic development, about the 5th week of development, so while the embryo is implanted in the uterus and in the uterine wall, we start to see differences in whether that embryo is going to develop as male or develop as female. The Y chromosome around that time is kind of activating its genes. And now the Y really is a much smaller chromosome than the X. It has about 50 genes on it right now that we have modeled and understood. The X has about 100 genes. And if I draw the X and the Y, how they really look, what we really see is the Y is just part of an X. It is missing, we think from way back in the ancestors of mammals, it lost a piece and gained a gene for actually developing maleness. So I want to show you some of these different regions here. The tips of the Y chromosome where it says PAR. PAR is the pseudo, this means fake, autosomal regions. So they're like fake autosomes, and they're on both ends of the Y chromosome. They actually are homologous to the X in these regions. And during synapsis and crossing over during prophase 1, sometimes the PAR regions cross over. So I did lie to you a little bit when we looked at those pedigrees when we were practicing the other day. And I said they don't synapse and cross over at all. They do, but not for the genes we were talking about. Here I'm introducing there is a tiny bit of crossing over if in an individual that is XY, but not affecting the things we were talking about. In the middle of it, here, you see it's got SRY, MSY, and then I have the centromere, that's the little pinched in bit. I want to talk about the SRY and the MSY. The male-specific region of the Y chromosome contains genes that have to do with the developing as male from the embryo through adulthood. They do not recombine with the X. You can see it is almost the entire gene. The SRY I have mentioned a few times in different lectures. This is the sex-determining region of the Y chromosome. This is where we have the male development controls and the code for testes. It codes for a protein called testes determining factor. I didn't make this red. Kind of as a hint too, as you're reading this, you're like, holy cow, SY, SR. I highly suggest, as we start talking about specific genes and the expression of them, that you do not start by memorizing the short version. I made these red. On the exam, I will write out both the short and the long specific version of it. I suggest that you learn male-specific region of the Y, MSY. Don't just learn MSY, because these will start to muddle around in your head like you would not believe. They're just letters. Turn them into meaning as you study them. MSY is the male-specific region of the Y. Once you have that down, now you can call it by its short name. Sex-determining region of the Y, the SRY region, is determining whether that embryo develops as male. This picture up here is actually just a picture of the Y chromosome. The SRY, that sex-determining region of the Y, we actually have a pretty good idea of what it does and when does it act. If I have some crossing over that accidentally occurs, and this is we do it on purpose within mice, but we actually do see it in some of the human population. There's always a chance that you do some crossing over between the X and the Y, and you're not just doing the tips of the Y chromosome. When that happens, I'll get a person who is XX chromosomally, but they develop testes because they have a piece of the Y, that SRY gene, stuck to one of their X's. They have the code for developing testes and all that male development, even though they're chromosomally have two X's. And that's why complete crossing over between those two chromosomes is a problem. If I did it all the time, if they tended to always cross over and synapse and go through the entire process that the autosomes go through, I wouldn't have any chromosomal tie to sex development. Transgenic mice absolutely have been researched and understood in a way that we can look at what happens to their Y chromosome. If I inject the SRY region into XX mouse eggs, so they have been fertilized, they have the chromosome complement XX plus the other autosomes for mice, those embryos will develop testes. If I take an XY mouse and I introduce a chemical that silences, silence means prevent the expression. If I silence the SRY gene in those mice, they develop as female. I did put, if this is your thing and you're interested in this, I did put 3 different articles down here. All of these should be accessible through the Camden Library. If you are unsure how to do this and these don't open, talk to me. I'll help you log in, but also I can link you up with the librarian because you're going to need to be able to do that as you go into producing your papers or presentations for the end of class. Let me do this. Let me just show you this real briefly because we'll look at this in a couple weeks. This is the result of something called gel electrophoresis, where I'm actually running genes and I break the genes apart and then I separate them by size. I run them through a gel, which acts as a molecular sieve, so it separates them by size, and then I can see that with my eyes. In this case, an XY mouse, if I separate out the Y, an XY mouse actually has the gene SRY. An XX mouse does not. If I take my mouse that was fertilized as XX, this mouse, and I add the SRY gene during development, the embryos will develop testes. These develop as male because they have the SRY gene. Transgenic means that I change the genes, I cross genes from one thing to the other. These are transgenic mice that I'm doing these experiments in. The SRY genes, the sex-determining region of the Y, encodes for a factor, a protein, a big one, called testes-determining factor. It's a transcription factor. So here you may need to think back to what you've learned about transcription and translation. When we start transcription in DNA, a bunch of protein ties to the promoter region, the start region, and then it transcribes. There's obviously control mechanisms in animals that turn those genes on and off, and I think in Bio 1 you don't talk too much about them. Now I'm going to introduce them as we go through the rest of the class. When that protein binds to the promoter region of the DNA during transcription, it often has to have other things in place that allow the transcription to go ahead. It doesn't just go. We're not bacteria. We turn things on and off based on what else is happening in the body. Everyone has the gene that is transcribed during the process of embryonic gonad, sex organ development. If you have SRY, so you have a Y with an active sex-determining region Y, it makes a protein called the testes-determining factor. That testes-determining factor binds to a different region of the genome, a different chromosome, and allows a gene to be transcribed that inhibits the development of female gonads. So I'm going to say it again, but in a slightly different way. If you have SRY, you make a gene that allows the transcription of a protein that prevents female development. This gene, we think, and we don't know, and I did try to find, this is taken from some of the papers on it. We think the targeting gene is Mullerian inhibiting hormone. Mullerian inhibiting hormone prevents the development of the Mullerian duct. The Mullerian duct is the duct that leads to the development of the female gonads, the ovaries, and the fallopian tubes, and the whole female sex development pattern. So every embryo starts out with the possibility of either being male or female. If you inhibit the development of female, you develop as male. If you do not inhibit or stop the development of female, you develop as female. And so this here, I've taken a little bit from a textbook. OpenStax is a freely available series of textbooks that are a little bit outside of the scope of what we talk about. But if you're interested in this and you want to look at this, I'm happy for you to do your presentation on it. So something to think on as we talk about that. So I'll go back 1 second. SRY codes for a protein called TDF, testes determining factor. Testes determining factor is a switch. It's a protein that binds to the DNA somewhere else, and it inhibits the development of female genitalia. That gene that it's grabbing onto and inhibiting is on chromosome 19, not on the sex chromosomes. If I look at anti-Mullerian hormone, so the cascade, the pattern of what the Y is telling the body to do, I see in females that are developing, so babies, that I see very low levels. I see very low levels of the hormone that is inhibiting female development. And that kind of makes sense, right? Because we're not inhibiting female development. In males that are babies younger than 24 months, look at this. I see massive amounts of anti-Mullerian hormone. The anti-Mullerian hormone is inhibiting female development. But the development is actually on chromosome 19. So we talked briefly when we talked about epigenetics and the fact that we could have control switches and epistasis. Those control switches can be switching on autosomes even though the controls are on the sex chromosomes. So you can already see the kind of questions I could ask you, which would be super, okay, I admit, maybe it's not fun, but interesting to think on. If you here have the Y, but fail to make anti-Mullerian hormone, what does the 19 code for then? And what happens to the phenotype of the individual that has that? We'd see their chromosomes different from their phenotype because we are not able to see whether the gene is activated or not. We can just see the presence or absence of the gene when we look at a karyotype. Overall, the sex ratio of humans, despite all that complexity, is about 50-50. That's a bit of a lie, 50-50-ish. Your book presented the data from 1969. I went and got it from 2024. If you've never been to Our World in Data compiles census data and other information from all over the world. It's a fascinating, you can rabbit hole that you could spend all summer on if you want. But the percentage of the proportion of the population that's female varies based on where you are in the world for many reasons. So we want to talk about the sex ratio, but then I want to talk about the sex ratio when you're born versus the sex ratio when you're an adult. There are more males conceived than females. So at conception, so during early development, when you're still in your mom's uterus, it's about 51 to 49 males to females, so more males. As the secondary sex ratio, the proportion that's actually born is actually closer to 50-50. By adulthood and beyond, there are more females than males. And so I don't have 100% commentary on this, but here, this color right here, and here to here is about 50%. And so it varies based on region, it varies based on what happens to you after you're born, but it does seem like more Y sperm are successful, but by adulthood that balances out. And then it keeps going the other way. So a little bit of practice with me. These are much easier, so give yourself a second, but no solving order of anything in this one. Really just, what does non-disjunction mean? In the first meiotic division, so meiosis 1, metaphase to anaphase, metaphase 1 to anaphase 1, what could the mistake of non-disjunction lead to? I'll give you a second. And then pause me if you need more time. I think you want to say A, but it's not A. If you said A, I want you to go back and think about what happens during meiosis 1. Do I separate homologous pairs or sisters? And I think it wouldn't be bad to go through meiosis one more time, probably many more times, and see where we would have two Y's stuck together versus where we would have an X and a Y stuck together. The answer here is really E. Klinefelter syndrome is XXY. Turner syndrome is X with no chromosome. The male would get these stuck together during meiosis 1. So he would pass on an X and a Y. During meiosis 2, the sisters would separate from each other. But during meiosis 1, the homologs separate from each other. Question 2. What describes human sex determination? Females are heterogametic. Individuals with a Y chromosome are male. And this is asking you sex determination. X chromosomes are female. Individuals with at least twice as many X chromosomes as Y are female. We'll bring that up. That happens in some things, but not in humans. Individuals with one X chromosome are male. This here is the answer. You could be XXY. You would still develop as male. This is not true in humans. One more. No, that was it. I have 3 at the end of this part. I will wrap up the video there. We'll go on to part 2, which is talking about some other mechanisms for sex determination. We'll bring in the turtles because I love them. We'll bring in some bees and talk about how some animals that aren't humans determine their sex. And I want to talk about bar bodies, and I have a video for you to watch. I'll link it in the little overview page that I think will help you understand bar bodies. And then I will see you in the next video.